chapter 5, and it is talking about biogeographical regions. And the first question that came up is, what is a biome? This is a large area that contains similar types of climate, soil, vegetation, structures, regardless of what type of species is currently living there. And we look into what a biogeographical region is, and it's a place in the world that has similar species composition, like from plants to animals. Next is, how are the boundaries of biogeographical regions outlined? Usually they're defined by what types of species are either absent or present at the time. So we have to look into if the species was born there, if they moved there from dispersal, or if they were absent within the length of either extinction, or if they were never even there. Okay. Explain one of the metrics used to quantify species similarities between pairs of location. One type of metric we used was the Jaccard index, which initially counts the similarities and compares the number of species between two different locations. So what they do is they take two different or a type of species from two different locations, and they have to count the totality of every single species that is there in order to compare what is going on there. So then we learn to describe the general patterns of species similarities in South America. Why are those patterns like that? In South America, their patterns of species are usually similar because their regions of land are usually flat or stable, and it allows their species to move freely. If we were to compare it to the Pacific coast, we have mountains, so therefore the mountains create steep types of environments, different areas, and initially will isolate the types of species that are there, so they can't disperse as freely as they would like to. And with that, we learn how can the use of higher taxonomy influence the boundaries of bioregions. When we use higher taxonomy like Genera, we compare two different regions being similar, even though there are different types of species there. It produces different boundaries depending on what level is used. How can the use of different taxa or species groups influence the boundaries of bioregions? We have to think that species are all different. They can leave, they can disperse differently. If we use that as an example, we can use birds. They can literally fly. They have little to no barriers other than possibly being eaten in the wild, or being hit or something, or crashing into something. But if you compare them to mammals, they usually can't move as freely because of course they can't fly. They can only jump and move so far and fast. So in general, each taxonomic group is able to produce a different type of map of bioregions. Let's see. Our last few questions. How can climate influence the boundaries of bioregions? Climate is always shifting, so the bioregions change because we have different species that have to migrate to different places. And if it's too cold, they're going to go somewhere warmer. Of course, if it's too warm, they're going to go somewhere colder so that they can adapt and they can continue to live over time. And humans, how can human activities influence the boundaries of bioregions? We tend to alter and change everything. We seem to ruin things before they can recover and fix themselves through habitat destruction, over-harvesting, climate change, and invasive species. We, as people, influence it because we overuse things and we don't give it time to recover. And our last question is, why are there so many different maps with different bioregions? They're different because every type of bioregion or biomap that has been created by someone else. There's different species, there's different scales and methods that people use in order to find the species distribution. So that is why there are so many different maps. And that is a conclusion to chapter 5. Thank you.